Hi guys, today we're solving Cambridge A-Level Biology, Paper 1 Topicals, Mitotic Cell Cycle. This is Chapter 5. The first question shows us a diagram of mitotic cell cycle, and they ask you which phase is DNA replicated in. So let's look at what we've got here. So we know that all of this is interphase. This is our mitosis, and DNA is replicated in S phase. So this is the S phase synthesis where DNA is replicated. Question number 2. Bacterial cells divide by a process called binary fission. We're talking about bacterial cell. Binary fission, we know what that is. Macromolecules, which macromolecules must be synthesized for this process? So our options here is cell membrane, proteins, and RNA. This must be synthesized because you're going to need it for new cell structures and the metabolic processes. So DNA and peptidoglycan, this is going to be needed as well. For the DNA is for replication, so the DNA is replicated for offspring, and the peptidoglycan is for building the new bacterial cell wall. Enzyme and cellulose. Enzyme we do need, but cellulose is going to be this is for plant cell walls, not for bacteria, so we don't need that. Our answer is going to be B. Question number 3. Which statement about the behavior of chromosomes during mitosis is correct? Behavior of chromosomes during mitosis is correct. Sometimes they can ask for incorrect, so just look out for that. A is they attach the spindle fibers to contain them within the nucleus. That's not gonna happen. This bit would be wrong. They actually attach the spindle fibers to because over here the nuclear envelope is gonna break down. And the attachment to spindle fibers is just to move them. B is they condense to prevent further translation of genes. So they do condense, but further translation of genes, that's not happening. C, they reach the poles of the cell and begin to uncoil. This is going to be our answer. This will happen during telophase, and this is when chromosomes will reach the opposite poles and revert to their chromatin state. Now, why is D not our option? Because they replicate to produce sufficient DNA to form 2 new nuclei. So replication is not going to happen here. They don't replicate here because DNA replication is during interphase. This is an electron micrograph of a group of human chromosomes. Let's label what we've got here. W is the centromere. As you can see here, it's where it's joining 2 sister chromatids together. So this is one and this is the other one. It's joining the 2 sister chromatids together. That's the centromere, W. X is here. and that's going to be your telomere. The description for the telomere is going to be that it's a protective cap-like structure found at end of chromosome arms. So it's going to be found here, here, and then here. So you'd find 4 in this one case. What's Y? Y is going to be your chromatid. So let's match them here. W is our centromere. X is telomere. And Y is chromatid. That's going to be C. One more thing to clarify is centriole, centrosome, centromere. What's the difference? When two centrioles are made up of microtubules. Two centrioles at a right angle, they make a centrosome. Centrosome is where micro spindle fibers are arranged, organized, and then that's one thing, centriole and centrosome. Now, centromere is a complete different thing. You'll have to excuse my drawing. But when you have the two sister chromatids, they are joined at the center by a centromere, like here. Five, the enzyme telomerase prevents loss of telomeres after many mitotic cell cycles. Which cell transcribes a high concentration of the enzyme telomerase? Enzyme telomerase comes up in a lot of questions, so it's important to know what it does and how it does that. This is going to be the enzyme that maintains the telomere length. And what that's going to do is it's going to allow the cell to divide repeatedly without losing the genetic information. And because of this function, it's going to be highly active in cells that need to divide a lot and divide rapidly. Think like cancer cells. Number 1, neutrophils. Neutrophils are type of white blood cell, a type of phagocyte. They're differentiated white blood cells. What we know about them is that they're short-lived and they don't undergo much division once they're mature. Because they don't divide much, they're not going to need this enzyme. Number 2, mature red blood cells. What do we know about red blood cells? Red blood cells don't have nucleus and DNA, so they don't divide or really produce this enzyme. They're not able to because they don't have the DNA. So this is not going to be. Number 3, activated memory T lymphocytes. What do we know about lymphocytes? This is from chapter 11, but lymphocytes are going to be of 2 types. We know T cells and B cells. T cells can again be of 2 types. We have T killer cells and T helper cells. But all we need to know here is that T lymphocytes, they are immune system cells and they undergo, remember, all of those, they undergo clonal selection and clonal expansion. So clonal expansion means they're going to be dividing a lot and they're going to need high levels of telomeres. That's the question, high concentration of telomerase. Sorry, not telomeres, telomerase. 3 is the only one that's dividing a lot, going through clonal expansion. So this is our only option, D. Question number 6, at which stage of mitosis do these events occur? Centromeres separate. We know that the sister chromatids are pulled towards the opposite poles of the cell. This is during anaphase. And chromosomes become shorter and thicker. This is condensation. This happens during prophase. So this and this, B, is going to be our answer. Moving on to the next page. Which row shows the correct number of each component of a single chromosome during prophase of mitosis? Okay, so during prophase, what we know is that there will be two sister chromatids joined at one centromere, and each chromatid is a DNA double helix. So there will be four polynucleotide chains. Four polynucleotide strands, and that means there will be two per chromatid, and every chromatid will have two telomeres at the end. So there will be four total. So I've written down everything here. Centromere, there is going to be one centromere joining the two sister chromatids, and chromatids, there's going to be two chromatids. There are two sister chromatids joined at the centromere. The polynucleotide strand is going to be four. There's two per chromatid, and then the telomeres is going to be four, one at each end. So B should be our answer. Question 8. The cell cycle includes mitosis. You know that there is interphase that has G1, S phase, and G2. Then you have mitosis, and then you have cytokinesis, which are features of nuclear division. So nuclear division, they're talking about mitosis. You know that it's going to be PMAT. You have prophase, metaphase, anaphase, and telophase. Form cells of equal size to the parent cell. This is going to be cell division, not nuclear division. 2. Forms genetically identical nuclei. This is correct. 3. Semi-conservative replication of DNA. Now this happens during S phase of interphase. And as we saw in question number 1, all of this is interphase, and this is completely different to this mitosis. So S phase is completely outside of mitosis, the nuclear division process. D is our answer here. That's 2 only, not this. Question number 9. Which process that occurs in cell division are represented in the diagram? This is the mass of nuclear DNA in a cell, and over time we're going to look at how the graph goes. So what processes would occur here? You have this bit where the mass doubles from 2 to 4. So this is your DNA replication, where the DNA doubles in mass. And then this is likely to be your G2 or early mitosis. And this is a nuclear division because the mass per nucleus is going to become half. And this graph is showing you the mass in a cell. So that's gonna decrease here. And then over here we can see that it's gonna become, this is where the cells divide. Cytokinesis happens where the mass, the DNA is going to, so the mass of nuclear DNA is going to return to the original levels. It's going back to 2 in one cell. So each cell is going to have it at 2. So it's going to be B, this answer. Everything else is missing something. So that's the only one that's right. On the next page, we've got the question, what happens to telomeres during mitosis of a cancer cell? So telomeres during mitosis of a cancer cell. So what we know is in a normal cell, it's going to lose the telomere length each time that it divides. But in a cancer cell, there is an enzyme called telomerase that we learned about earlier. It was in a question here. This question talks about that enzyme, and what this enzyme does is that it actively rebuilds our telomere. So this will allow the cells to divide rapidly and a lot of times without losing the genetic material. So what happens here is that the length doesn't increase, it does not decrease either. It's not completely lost, it's going to stay the same because of that enzyme. The graph shows 3 measurements obtained after metaphase of mitosis. So after metaphase, that's going to be anaphase and telophase. Let's look at the graph. We've got these 3 lengths, 1, 2, and 3. And they ask you what measurements do the curves represent. So the distance between centromere and poles of spindle, distance between centromeres of sister chromatids, distance between poles of spindle. So what we know about the distance between centromere of sister chromatids, centromere is what joins the sister chromatids together. This is going to be joining them at first, and it's not going to have any distance until they're pulled away during anaphase. So this is then going to increase. So they're going to move further apart. So that's 0 here. And then that distance is going to increase. So that's going to be 2. And the other one, distance between poles of spindle, this is already going to be high between the poles. There is already going to be some distance, and then it's going to increase further. So it's going to be this one, I think, because the distance between poles of the spindle, so after metaphase is anaphase and telophase. This is where the cell is going to elongate, and it's going to push the poles further away from each other. So this is our 3 is our answer for that. And the distance between centromeres and poles of spindle is going to be 1. And let's look at it. What's happening here is it's decreasing. That's the distance between centromere and the poles of spindle, because in anaphase, the chromosomes are pulled towards the pole, so that distance decreases, it shrinks. So it's going to be 1. And that's A as our answer. Next question. A scientist stains the chromosomes of a diploid plant cell with a fluorescent dye to observe the telomeres. This cell has 38 chromosomes. How many telomeres, the protective ends, so it's going to be 4 on each, in case of when you have 2 sister chromatids together. But let's see what's happening here. How many telomeres will the scientist observe in one of the nuclei during, so this is in one of the nuclei during telophase of mitosis. This is when they've already been pulled apart, the sister chromatids. So this is going to be, you're just going to take this 38 and times it by 2. 38 chromosomes times 2 telomeres at each end. That's going to give you 76 as your answer. But what's happening here is you have, so each individual chromosome, it will have 2 telomeres, 1 at each end. And at telophase, this is important, each new nucleus contains the diploid number of cells. So that's important to know. This is what we were given. And the diploid number is of a single chromatid chromosome. So that's what we do. 38 chromosomes times 2 telomeres each. That's going to give you 76. That's your answer. Moving on, they've given us a photomicrograph. We can clearly tell that this is anaphase. You should be able to identify it because the sister chromatids are separated. You can see they're separated at the opposite poles, so they're being pulled towards opposite poles. This is going to be anaphase. One way to remember is in PMAT, which is for mitosis. P is prophase. M is for metaphase. So you can just think of them. They're aligned at the middle. So they will be aligned in the middle and at metaphase. So M for metaphase and M for middle. And then A is anaphase. So anaphase is away. A for anaphase and A for away. So they're going to be pulled away. Now, what is happening in the cell? Centrioles are replicating. This is not happening because replication, DNA replication, and centrioles replicating, that happens during the S phase of interphase. So this is mitosis that we are talking about here. So this is not going to happen. Spindle microtubules are shortening. Yes, they are going to shorten because they shorten to pull the individual chromosomes towards the pole. And chromatin is condensing that happens during prophase. By anaphase, it's already condensed, so there's no condensing happening here. It's 2 only. This one. Which events listed are part of mitosis? Interphase is a complete different thing, so this is not going to be part of mitosis. Interphase is when G1, S phase, and G2 happens. Prophase is part of mitosis. Cytokinesis is going to be after mitosis, so this is not happening in mitosis. That's D. The next question is matching parts of chromosome to their functions. So these are the parts and these are the functions. Centromere holds the coils of DNA together. That's not centromere, that's going to be the histone protein. The histone protein is P2. P2 should be matched with this, and then holds 2 chromatids together. That's going to be centromere. And telomere prevents loss of genes. That's correct. So let's match them. So the only accurate pair given here is going to be P2 and F1. That's this one. So B is going to be our answer. The next page. The graph shows the length of the spindle fibers during mitosis. Which region of the graph shows when all the centromeres have detached from the spindle fiber? So let's look at what the graph is showing. This is time against the length of spindle fibers. This is when the spindle fiber length is increasing. That's going to be in prophase. And this is when the spindle fiber is growing in prophase. B is when this looks like when it's reached its maximum length and it's going to be attached to the centromere. This is metaphase. This is going to be at the equator of the cell in the middle, like here. And you're going to have to excuse my drawings. You have to bear with it. Now, this one is going to be anaphase because the spindle fiber is shorter as it pulls the sister chromatids towards the opposite poles. We talked about this in an earlier question, but it's still going to be attached to centromere during this phase. Over here, the length is really decreasing. The spindle fiber is going to finish its job here, and it's going to be completely broken down. This happens in, we know that this happens in telophase. And once it's completely broken down, obviously it's going to be detached from the centromere. So our answer is going to be D. You can read through these if you want. 17. The enzyme telomerase prevents loss of telomeres after many mitotic cell cycles. We've done a couple questions relating to this already. Which cells need to transcribe, this is slightly different. So which cells need to transcribe telomerase enzymes? So which one will be transcribing them? Once again, enzyme telomerase, this is required by cells that divide extensively, or like they divide rapidly. So they're going to be dividing a lot, and then they're going to need this enzyme. 1. Cancer cells, we've been going through this. Cancer cells for sure is going to have this enzyme because cancer cells will be dividing rapidly. They divide uncontrollably. We have stem cells. This is also going to need it because stem cells must divide to differentiate and in order to replace the differentiated cells. And activated memory B lymphocytes, this is also going to need this enzyme because activated memory B lymphocytes, when the immune system is triggered, they undergo rapid clonal selection. That's one thing, but clonal expansion is what's relevant here. Clonal expansion is just like many rounds of mitosis, so they're definitely going to need this enzyme. So all 3 of these. That's going to be A. Next, they've given us a diagram that shows some of the stages would take place during the cell cycle. Okay, what are they asking? Which 2 stages take place during interphase? So we know interphase is G1, S phase and G2. The number of DNA molecule in cell doubles. This is DNA replication. This happens during S phase of interphase. Cell grows, this happens in G1 and G2, which is again part of interphase. And 3 chromosomes become visible. This happens in prophase, which is part of mitosis, so not this. And 4 is nuclear envelope disappears. This happens again in prophase, which is going to be in mitosis. 1 and 2, that's A. 19, the enzyme, again on that enzyme. The enzyme telomerase prevents loss of telomeres after many mitotic cell cycles. Which cell needs to transcribe telomerase enzyme? Stem cells, we already talked about this. This is definitely going to need it. Activated memory B lymphocyte. Stem cell, because they must divide to replace differentiated cells like we talked about earlier. 2 is activated memory B lymphocyte. This is again when immune system was triggered. They have to undergo rapid clonal expansion. So many rounds of mitosis, so they're gonna need it. T, helper T lymphocytes secreting cytokines. Now this is gonna be slightly different from the previous case because this is helper T lymphocyte that are secreting cytokines. This is already differentiated, so this is not going to be dividing because it's already differentiated and it's performing its functions. This is one that you might think is also going to be part of the ones that need to transcribe the enzyme, but no, this is already differentiated. It's performing its function, that secretion of this, so it's not going to be needing that enzyme, only these 2. That's B. Moving on to the next one, we have a diagram of a cell that's showing during mitosis. So what stage does it look like? This is anaphase because you see anaphase, A for anaphase, A for away, is being pulled away. You can see they're being pulled away at the opposite poles. This is and they want us to identify whether it's a plant cell or animal cell. It does not have double lines showing that there is a cell wall, so this is animal cell. That's C. A gene codes for the production of protein P53 that binds to damaged DNA during interphase and prevents its replication. A carcinogen in cigarette smoke mutates this gene. So this is going to be our trigger. The carcinogen is cancer causing. It's going to mutate the gene. And what this gene does is that it binds to damaged DNA during interphase to prevent its replication. So to make sure that damaged DNA is not being replicated. Which statement explains why this mutation may cause cancer? Cells with no PF3 are allowed to undergo mitosis. This does happen, but this is not really what they're asking. So B is cells with no PF3 allow their damaged DNA to replicate. That's the problem here. So this is going to be our answer. Why not the other ones? The carcinogen in cigarette smoke increases the rate of cell division. This is the carcinogen, this is a trigger, but that's not telling you why the mutation may cause cancer. So this is just like the trigger that leads to this. The PF3 causes uncontrolled cell division. It does not cause uncontrolled cell division, so that's wrong. During mitosis in animal cells, which process occurs after prophase? So after prophase, you have metaphase, anaphase, and telophase. So we're going to see which one describes it well. We have centrioles move towards the poles of the cell. This is going to be prophase. So this is when the spindle fibers begin to form. The centrioles will move towards the poles of the cell. So this is happening at the very beginning, not after prophase. That's our question. Next one, centromeres attach to spindle microtubules. That is right. This happens after prophase. So this is going to be our answer, but let's see why not the other one. Chromatids join to form chromosome. This does not happen. The DNA replicates during S phase, so what even is this topping on? So this is not going to be the one. Chromosomes condense and become visible. Condensing, this is again prophase, so not the answer. We can do a few more questions before I end the video. Embryonic stem cells are able to replicate continuously. So that's going to be embryonic stem cells. It's going to be similar to cancer cells. And remember, cancer cells, they need that enzyme telomerase. What happens to the telomeres during, they're also going to have that enzyme. So what happens to the telomeres during repeated mitotic cell cycles of embryonic stem cell? So what will happen, similar to the case of cancer cell, the length is going to stay the same because they have that enzyme. This case where the length decreases, this happens in cells that lack telomerase enzyme. 24. How many copies of each DNA molecule will be found in a cell at each stage of the mitotic cell cycle? So at G1, this is before S phase. This is not going to be replicated yet. So it's just going to be a single DNA molecule. And at this stage, this is after mitosis, it's going to already have passed the S phase where DNA was replicated, and mitosis where the nucleus divided into two nuclei. And then it's going to be 2 here. Next question. Which metabolic processes will be very active in a cell that has just completed cytokinesis? ATP formation, yes, because there's going to be a lot of growth and synthesis of new organelles, so you're going to need ATP energy for that. DNA replication, this is not going to happen because this happens during S phase. So this is not going to happen straight after the cell has divided into two. Protein synthesis is going to happen because after cytokinesis, there'll be G1 straight after in the whole cell cycle. So once it finishes that, it's going to move on to G1. This is when the cell is going to be highly active in transcribing and translation to build enzymes and stuff. So there's going to be protein synthesis happening. 1 and 3, this is B. Alright, last question for today. A mutation occurs in a gene which prevents the production of telomerase. This is the enzyme because it ends with ACE. That should be your clue. What is the effect of this mutation if it occurs in bone marrow stem cells? What's going to happen if there is no enzyme that prevents because of this mutation, there won't be production of this enzyme. And what's the purpose of this enzyme? It prevents the loss of telomeres. So bone marrow stem cells, it will keep dividing. So what are our options here? Rapid increase in the production of lymphocytes. That's not happening. A tumor grows in the bone marrow. Tumor does not grow. Bone marrow stem cells eventually no longer divide. This should be our answer because this is just going to, the telomere is going to keep shortening every time it divides, and eventually it's going to stop dividing. D, why is that wrong? Because the total blood cell count will be unchanged. It won't be unchanged because if bone marrow stem cells, which produces all the blood cell, differentiates into blood cells, if that bone marrow stem cell stops dividing, then the body is eventually unable to replace the blood cells, and the total blood cell count is going to drop significantly. So this is not going to be our answer. It's going to be C.